Hi, everyone. My name's Joe. I'm from Miltenyi Biotec. Thanks for tuning in to our webinar this afternoon, or I guess this morning. I lose track of time in this room. In any case, I'm going to be talking to you a little bit about our instrument, the MACSQuant Tyto, and how it has supported some groups making the transition from normal standard laboratory work into an infectious hazardous material type application. It seems like there's a lot of people making that migration with rapid accumulation of COVID research. And I think we have a really unique platform that allows for users to feel safe with their operation on the device in a traditionally quite open system type of technology. Myself, I'm from San Diego area. I went to graduate school at UC San Diego and did a lot of cell sorting on an influx device. This is a traditional jet and air sorter. Spent a lot of time in a small windowless room with that device. And after my PhD, came to Miltenyi and have since learned a lot about where cell sorting fits in the grand scheme of the science community, and have had the opportunity to work with a lot of experts in a variety of different fields. Today, we're going to focus mostly on the biohazardous element and kind of that arena of cell sorting. And I think that's particularly applicable to the COVID-19 landscape. So but with that, I will go ahead and jump into this. From an agenda perspective, I want to kind of go through the challenges with this type of work and really the advantages of using something like cell sorting technology, but it does come with caveats. I'll give you a little bit of background on our sorting instrument, the Tyto, and then talk about how a couple of groups have used that instrument and validated it for work with hazardous materials, and then kind of go over a couple of applications that they've actually been able to run with it. So I'll look to stay on time as best I can, and hopefully go through this in a manner that even if you're new to the technique or new to the instrument, that we can hopefully have everybody pick up something unique. So in the interest of that, I'll start very, very broad on really cell enrichment techniques. And I think anybody who's working in a cell biology field, immunology, you've done some sort of cell enrichment, and that can be as trivial as an antibiotic selection for an E. coli culture, or as specific as a 15-color panel looking for a very specific subset. There are drawbacks to each of these techniques, and but we'll go into a little bit more detail. As many of you may know, Miltenyi's real flagship technology is magnetic bead enrichment. That has started almost, it must be 30 years ago now, because it was 1989, I believe, that the patent came out. These are antibodies that are specific for antigens on cell types. They're conjugated to iron oxide particles for magnetic enrichment. So we can pull out specific cells from a co-culture and use those cells for downstream assays. And this is a pretty much ubiquitous technique now, and it's led to a pretty amazing advancement in clinical applications in cellular therapy as well, which is really quite exciting. This technique of magnetic enrichment has the advantage of being able to be done in a closed system, which has obvious implications for cellular therapy and sterility. But in the context of today, it has particular benefit in that it contains the material and allows an operator to feel a lot more comfortable working with technology like that. Historically, something like fluorescence-based cell sorting or flow cytometry sorting hasn't afforded that luxury. These are usually done in an open environment, something that requires the cells to be passed through the air to be sorted effectively. And really, of all the flow-based sorters that are in the field currently, it's got to be in the high 90% of instruments that are using this type of technology. So that makes it really quite difficult to scale that into something that is really approachable for a new operator in this environment, because the list of requirements to feel protected with an aerosol generating system is a long one, and it's one that requires really quite a bit of expertise to feel ready for. So I'll go into a little bit more detail on what that requirement is, but I think what we are going to be really focusing on today is not so much these bulk separation strategies. I think these are well conserved. I think people are generally comfortable with the idea of how these can be implemented in a environment with hazardous materials, but instead focus on flow-based sorting, flow cytometry sorting, and how this technology has grown and adapted to fit into this environment. Now, I should say there absolutely are methods that use the current predominant technology, that's jet and air sorting, in a high BSL environment. So it's not to say that you can't achieve a safe operator environment with traditional technologies. I think, and the point I hope to make throughout this presentation is that it's simply very difficult to do that, or at least implementing that is going to be far more complex or involved. And I'd like to make the case that a different type of system will allow you to make that transition with a bit more confidence and a bit more ease. So I've touched on this a little bit already, but bulk cell isolation methods, they're very common. In general, you don't get a really great specificity of the cells that you're going to be pulling out, but they're incredibly useful for large amounts of cells. They're really quite easy to transition. I think what you gain from something in flow cytometry sorting is really a high parameter sort, something that allows you to be very precise about the cells that you pull through and very exclusive about the cells that you pull through as well. So that means any sort of contaminating cell type can be excluded quite nicely. Some drawbacks to that technique, I've already talked about the aerosolization part, which we'll go into more detail about, but they also require a quite trained operator to use these technologies. Many of you probably use a cell core or a flow cytometry core, and those are trained experts that spend a lot of time in the morning setting up their machines, a lot of time at night shutting them down, and then we'll be monitoring them throughout the day. So it can be a prohibitively difficult technology to take on into a lab that doesn't have the expertise. So in the interest of kind of getting everyone on the same playing field, I'll do the classic intro speech where you talk about the Webster's Dictionary for a hazardous sample. It's really what you think it is. It's something that has the potential to pose a risk to the safety of an operator, or something that might contaminate the system itself. And we'll see that both of those pose significant risk when you're talking about something as infectious as COVID, or as scary as some of these BSL-3 and 4 microbes that individual labs are working on. We're going to try and keep this focused more on the COVID-19, SARS-CoV-2 world, in the interest of staying on topic today. But these same sorts of ideas and technologies apply for hazardous microbes, or plasmodium, really pretty much anything that you would deem scary or hazardous. So quickly, I'll go into a bit more detail on what a traditional jet and air sorter does to separate the cells, and how we mitigate the risk to the operator. As I mentioned, this is something that is done. Plenty of cell cores, flow cytometry cores, operate with this type of technology, but the infrastructure to do so is pretty significant. So a jet and air sorter, very briefly, the way that it works is it pressurizes the cells in a device. That pressurization shoots the cells out of a nozzle into the air, at which point cells that have the desired phenotype are they have a charge applied to them, which allow a magnet to physically separate those cells in the air. In the theoretical perspective, I'm always fascinated at how well these systems work. It's actually quite remarkable how far this technology has come from such a seemingly impossible type of engineering technology. So I've always been quite impressed with this. The difficulty here is obviously that you're spraying cells through the air in a space where humans are trying to do their science as well. So the way that that's mitigated is whole sorting systems are placed inside of biosafety cabinets, which can help minimize the amount of aerosols that actually make it into the room. These cabinets also will have specialized air handling systems or aerosol capture systems. Operators themselves can be required to wear ventilation systems. This is something everybody's quite familiar with now. And really the part that always was more of a mystery to me is the decontamination of the instrument and the housing. If anyone has spent a lot of time with a cell sorter, this type of cell sorter, you know that a clog that goes through can spray material all kind of inside the device, which poses an outrageous amount of work to get that clean. And that's a person who has to go in there and do that cleaning. So under those circumstances, somebody's having to either take on that risk or be abundantly cautious when cleaning out these systems. Obviously, microbes in shared fluidics lines pose the risk to contamination of future samples, but I think it should be fairly well understood that working with a system that deliberately generates aerosols of hazardous material just doesn't feel good. And so I think that's all I'll say about the systems for now. I think I've made my point there. When COVID-19 outbreak occurred, I follow a message board online that's contains a lot of the flow experts from around the world. And it very quickly became the question of how much caution do we need to take to run this material? And I've followed that with quite a bit of interest. Many labs are equipped to do HIV sorting and things like that, but with the excitement about working in this field, there are a lot of people entering into the space that may not have the same level of expertise or the same infrastructure. And so eventually we kind of settled on, the ISAC, the International Society for the Advancement of Cytometry, I believe is what it's called. They settled in on some recommendations for operating with COVID-19. The COVID-19 patient samples, they can be safely sorted in a BSL-2 laboratory with certain BSL-3 level practices in place during the sort. In general, an aerosol management system and respiratory protection for the operator at all times when the cell sorting chamber is opened, and an SOP detailing procedure in the event of a nozzle clog. So I've already gone into a little detail on these things. This is the recommendation from the NIH and the ISAC Biosafety Committee. So this is as real as it gets for how we've been asked to operate under these conditions. I think this is an obvious thing to implement for groups that are deliberately working on COVID-19 samples. I think it's a reasonable practice in labs that are working with human material to operate with some level of caution or assumption that it's possible that infectious material is present in any blood sample. So as much as I'd like to think that every sample that comes from your blood donor facility is clean and happy and healthy, unless you're actually testing for that before it gets into your facility, taking precautions like these is probably safe. So here's a little bit more granularity on these details. We want to test containment of the pre-sort using a Cyclex D procedure. I'll go into a little bit more detail on this as we talk about aerosolization. This is a test to verify that your system is able to contain aerosols and minimize that spread into the room. I've also highlighted a couple of other things, checking the room for negative airflow, Tyvek full body suit, gloves, and shroud with HEPA filtered PAPR to be used at particular times during the sorting procedure. So really, you can imagine operators are gowned up quite substantially, and this needs to be done in a room dedicated to this type of research. So I hope this is becoming more clear that it's not a trivial setup to get these things moving. This is from the ISAC president and from the University of Pennsylvania Medical School. And let's see, what is the particular point I want to make here. Many of this is deliberately set up such that you have minimal operators working in the actual room. But there are parts of working with these instruments that require somebody to be physically next to the machine, but the operator will obviously have all this PPE, but in the interest of not gowning up everyone, usually in this case they have asked the individual who brought the sample in to sit in a remote location and guide the sorting experiment from elsewhere. So there are systems that have been put in place that help make this work, and they are mostly based on strategies that have been developed for working with HIV and other infectious diseases. Okay, so I think I've made this point already, but just so that it's very abundantly clear, flow cytometry is a hands-on, and it requires people to be involved, and jet and air sorters are potentially hazardous due to their requirement to produce aerosols to function. So we're really looking to find some other technique to do flow sorting, something that gives you the same benefit of multi-parameter sorting, but allows you to do so in a fashion that can be implemented in a facility that maybe wasn't originally designed to do that. So obviously this is a good opportunity to bring in the MACSQuant Tyto Cell Sorter. This is Miltenyi's product. We collaborated with a group called Owl Biomedical that is based out of Santa Barbara. We've since moved all of the device manufacturing to our Germany site, and this is now a full Miltenyi product. The Tyto is a really exciting machine, and when I joined Miltenyi a few years ago, this was really what brought me to the company. I really just see a lot of elements of this that are quite transformative to the field. I've spent a lot of time on droplet sorters, and the amount of the things that bug me about droplet sorters are really addressed with this system. So quickly, I'll try and do a little side-by-side on how these things are different. The MACSQuant Tyto is, let me actually quickly go back. The device on the right here, the instrument, is a cartridge-based system. And the way that it works is you would add your material into a cartridge. So generally, and particularly in a hazardous sample or something you're meant to keep sterile, any addition to the cartridge will be done in a hood, a very commonly closed situation. That cartridge, once it's closed, is presumably safe to use. And I hope I can make the case that we don't see aerosols coming off of this cartridge at all. So handling this outside of the hood is something that we've deemed is certainly better to work with than an aerosol device. So once the cells are placed into the cartridge, they are then dropped into the cell sorting device, the Tyto. This is a benchtop instrument. It's really quite a small footprint compared to an enormous biosafety cabinet with a sorter inside of it with the fluidics that are attached. There's no onboard fluidics. Everything, the only liquid is really what you see in the cartridge on the left side there. The system's microfluidic based, and it will use 3 lasers and up to 8 colors to differentiate cells into subpopulations that you may find of interest. And then when you designate which cells you'd like to use, it will pass through a microfluidics chip into your positive sort chamber. No waste is generated. Anything that's not sorted goes into what we call a negative collection chamber. And I'll show you a little bit of the benefits of having the ability to hold on to that material as we go on. So in the context of how this differs from conventional sorting, these cartridges are single use, so there's no carryover from sample to sample, so you don't risk contamination of your current samples with something you sorted previously. Everything stays in this closed system. We're not having to apply a charge to cells or launch them through the air, so that it's generally considered safer to cells. And we have quite a bit of experience investigating downstream assays that are, I think, quite compelling to that statement. And really, in the context of this environment, keeping the cells in a closed chamber is really advantageous. And once the chamber is placed into the Tyto, it's a hands-off sorting procedure. Anecdotally, I've run this machine, drove into a facility, took the Tyto off a truck, set it on the bench top, had it sorting within an hour of the truck parking in the parking lot, and then ran a presentation while the system was sorting and came back to sorted material. So the ability to operate this machine without sitting there and monitoring it is incredibly valuable, especially if you are in an environment where you don't necessarily have a super trained flow cytometry expert. With respect to hazardous samples, the closed cartridge, I think, is easy to internalize why this is beneficial. We don't need special air handling systems or biosafety cabinets. But the cleaning is really potentially not even a thing, because the single-use cartridges can just be thrown away at the end. There's no internalization of liquid into the device. Everything stays within the cartridge itself. I don't know how much you can see on my camera, but this is the cartridge here. So they're definitely small. They're also robust. We did some testing with dropping these on the floor in a lab facility and checked aerosols, and still nothing or anything like that. So I think unless you have a step that requires you to bash your cells with a hammer, we'd really anticipate that your cells remain inside this chamber for the duration, and that incredibly minimizes the requirement for cleaning of the system itself. The cartridge has these lure locks on the top. This is familiar for people who've worked with sterile blood products or things like that. And the way that the system's pressurized is via air going through a 0.1 micron hydrophobic filter. And so that is plenty small to keep out just about everything. And I think in the testing we've done, I've been really quite impressed with how little or how sterile this really keeps the machine, or your samples, excuse me. From an operational perspective, I'd love to go into more detail about how much awesome tech this cartridge has, but in reality, all you do is take off the cap and put your sample in one side, and then put the cartridge into the Tyto, and it kind of does the rest. So we used to have all these slides on detail of the different cartridge elements, and they're really not that profound. I mean, there's a lot of the really exciting parts of the heart of the system comes in the microfluidics chip. So the cartridge itself is merely a vessel to try and get your cells there. And again, this is a single-use system, and it's the only liquid in the whole platform. So this is really quite nice. I mentioned plug and play. This is really, once you load the cartridge and put it into the Tyto system, it will automatically align for you. We have these marks in the microfluidics chip that allow it to align the laser spots. You see these 3 crosshairs, the red, blue, and violet across the middle channel there. Those are the 3 laser spots. And so as cells pass through this channel, they're interrogated by the lasers. The system will validate which cells are of interest, and then they open up a valve, which is located on the right side. We have lots of cool videos on this on our website that will allow passage of your cells of interest. So this is, there's no droplet break off that you're trying to stabilize. There's no maintenance or upkeep that you have to monitor. It's really, once this is set up, it's really quite stable. So again, this can be really quite operator free. Okay, so really, it's cool that we have this device, but how has it been used in research? That is probably actually what's interesting. This is a kind of a brand new paper that's come out from the NIH, and I believe it's the Rocky Mountain National Laboratory, which has gotten quite a bit of press lately. But their motivation for acquiring this device was being able to use a sorter that they felt confident about in a BSL-3 setting, and that would give them the opportunity to evaluate potentially doing BSL-4 cell sorting, which really isn't a thing in today's world. So this paper, I think, it's a quite simple paper from a science perspective, but it shows that what the common problems are with the cell sorting in this environment. I've already gone into a lot of detail there, but briefly I'll mention which ones they were particularly concerned about. This is paraphrased from the paper itself, that a lot of times this material is hard to come by, so loss of cells is particularly damaging as a limitation of a traditional sorter. Aerosols are obviously a big problem. And any additional needs would be a sterile sorting process. So anything downstream you can be confident is going to grow out without contamination, and then easy maintenance and decontamination. So obviously in these facilities, anything that's coming into contact with a hazardous material can be potentially hazardous. And so everything needs to be disposed of. So systems that run on a sheath solution that, you're going through liters and liters in the process of setup and shutdown and operation, all that has to be disposed of in some manner. So working with a system where all the waste you generate is this, that's really quite beneficial. So I've talked a lot about aerosols, but not how we test for them. So this is a very common aerosol detection setup. The Cyclex D is the name of the disposable device for checking out aerosols. And basically they're these yellow-green beads, 1 micron in size, that they use to qualify if these are escaping from a particular device. These pictures are taken from a draft, like an internal setup that we tested at Miltenyi. This is, I believe, going to be published somewhere, maybe from a poster. Basically, in short, this was set up on the Tyto, and we ran a few experiments, and really we couldn't detect any aerosols being formed at all on the Tyto. And in fact, it was so few that we actually had to use an atomizer to generate enough of these beads to actually get a signal, so we could determine that the instrument was, or that the Cyclex D testing was even working properly. So in the positive control, we can definitely see these red circles in figure B, indicating beads, and in the positive or in the negative control, we see no beads. This is as expected. And then in figure D, this is our actual experiment. We don't see any beads as well. And this is nice, but the caveat is always like, well, yeah, Miltenyi did this experiment, so why would we publish I mean, as a scientist, I hate that response, but this is the reality of working at a company now, is people always have reservations about our own data. So it felt really good when the Rocky Mountain National Laboratory ran a very similar experiment in their facility, trying to replicate this data. And they took fluorescent beads, and very similarly, they ran them on the Tyto and on their current system, which is a droplet-based sorter. In all fairness, the aerosolization mitigation, I can't think of the name, the AMS system was pulled off of the ARIA. So I don't think this is a totally fair representation of aerosolization on the ARIA. But the point that I like to point out is that really in testing on the Tyto in a 10 minutes placed inside of the actual device, we see absolutely zero beads being captured. And this was tested across the entire cover slip. Obviously, there's just representative images here, but the entire cover slip was checked on the Tyto ones, and it did not show aerosols. So I think this is as good a proof as any. I'm convinced by this data. The group also, as part of that paper, ran a trial test on sorting something quite basic, pulmonary T cells. And I won't go too much into the detail here, but I think it shows that they were able to sort well within their expectations or normal parameters for sorting. And interestingly, when comparing activation of the T cells, which is usually a response to other cells present or some other stress involved with the sorting or other application or assay process, the Tyto sorted samples actually had lower activation than those purified with magnetic beads, which I think of as being particularly gentle already. So they checked this back to probably being due to not having quite as pure of a sample that came out from the magnetic particles, but just showing the power of having a more specific sorting device or something that allows them to get to a more pure final product. This is probably the most exciting part, is we've started to implement this device into facilities that are actually looking to do COVID-19 sorting. And I want to point out a group at University of Chicago who have a Tyto Cell Sorter, and I'll actually just read through this part here. I have to give a plug for a future webinar that's coming up soon. We have some of this data in-house, but I was told not to present it right now, and instead to let a member of Patrick's lab do the actual presentation there, in the interest of kind of having an expert talk about this data. And so that data can be found in an upcoming webinar. Please reach out, send me a message if you'd like to have an invite to that webinar. We are happy to have people join that, and it will be displayed in a context that I think is done by the right person to present it. That said, I'll read quickly through his talking point on using the actual Tyto. We have used the MACSQuant Tyto Cell Sorter to sort B lymphocytes from blood donated by individuals who have recovered from COVID-19. We found the Tyto much quicker and simpler to use than traditional cell sorters. Furthermore, the sterile cartridge allowed us to safely sort our BSL-2 plus materials at our campus's flow cytometry facility without any issues. The low pressure and gentle microchip sorting mechanism maintained high cell viability, which enabled us to easily perform 10x genomics on very rare populations of cells. Obviously, this is a pretty generic statement on the Tyto. I really think most of what he's saying is everything. It confirms what I've seen in the field as well. And seeing research like this come out is always redeeming, because I know what the system can do, and seeing it in the hands of people that are experts in the immunology side or in the infectious disease side is always a great feeling. So again, quick plug, please reach out to me. I will get you connected to the actual data that's come out from this experiment, and I'm really quite excited by it. All right, so now getting into another group that's used the Tyto in a COVID-19 context. This is a group in Denmark that has used the Tyto to work on an antibody treatment, and they have used it in a similar context. I think this slide is so interesting, the picture that they've put up here, because there's sort of a gut reaction to seeing people in a lab facility without gloves, without a hood, without masks, working on a sorter that's sorting infectious material. And there's a lot to be said about that I think the instrument has provided enough confidence to them that they know they're working in a contained device, and that was deemed to be safe for them to work in even a BSL-1 setting in Denmark. So this is something you'll want to talk with your EH&S about for sure, but I think there's precedent to believe that this sterility and containment is really quite different from what we would expect on a normal cell sorter. All right. These are statements from an interview with this group that Miltenyi conducted, and we were curious about, are the B cells themselves actually even hazardous anymore if a patient's recovered from coronavirus? And so they basically just treat them as if they were infected still. So that means transitioning this work from actively infected to post-infection doesn't mean that they transfer their hesitation on working with this material to a lower level of containment. They really still consider these as hazardous even after the patients are recovered. So we can imagine there's going to be a lot of individuals who've recovered from this disease that will have interesting medical questions to inquire about, and I think there's reason to be cautious when working with any of that material. Okay, let's get to this one. So in here they say they actually they feel safe moving samples from BSL-2 or even BSL-1 because the system's fully closed. The droplet formation systems need to be in a biosafety cabinet or BSL-3 due to aerosols. It really just changes the entire infrastructure of how they were able to do this type of research. And I'll be excited to see what comes out from their actual COVID research. So we've done this chat with them. They're kind of just getting going on Tyto. This lab is a HIV focused lab primarily, but with the world in the state that it is, they decided to switch to COVID research. I'll show some slides from the HIV side where they had developed some of the Tyto work, which I think will give some context into how they plan on using this in the COVID side as well. Again, safety is by far and away the major benefit according to them. It doesn't require in-depth training. This is particularly good for groups that don't have a lot of experience on cell sorting systems, and if you want to be as hands-off as possible. And then finally, there's no issue with containment and the operator burden of any sort of cleaning and decontamination is really a lot different. So with that, I'll move into some actual data on their HIV sorting. And real quickly, their experimental design was getting HIV positive blood from an individual, and this is not fixed material. This is actually HIV positive blood, and they would do a magnetic enrichment for CD4 positive T cells, and then staining for CD45RA and CCR7, and sorting on the MACSQuant Tyto. So I would say this is a pretty standard workflow for individuals investigating T cells in infectious disease. I think many of you probably have similar types of workflows for taking things to the flow core. So sorting these T cells out I mentioned earlier that there's no waste that's generated. So getting material that's from an infected individual is often quite difficult, and that material becomes very precious. So putting it through a cell sorter that will spit anything that's not sorted into a waste chamber that goes away forever is really undesirable. There are strategies that can help you minimize that, but here everything stays in the cartridge until you yourself pull it out with a pipette. So what they were able to do was sort multiple populations from the same material, and they did that by simply taking from the negative sort chamber and then placing that into the input side to sort out the next cell type. So from this, they were able to get effector Ts, central memory Ts, naive Ts, and terminally differentiated T cells from the same set of material. So that's just one blood sample. The first question I always get in response to this is, what about the viability of going through the chip multiple times? Viability after sorting is in the 99% on all of these. And this is something I've seen across a lot of different materials. So I'm of the impression that sorting through this device it's often more damaging for the cells to just be in single cell suspension at all versus going through the chip itself. So most of the time viability issues are due to the sample preparation side and not due to the actual sorting. And I think this is a pretty good indication of that. We were able to get great purities out of each of these, even for fairly low starting percentages. I guess 14% is not that low, but I think the next slide will hopefully shine a bit more light on that. So I think this data is meant to show that the Tyto is a comparable sorter from a utility perspective to what you're used to working with. I think as the field has seen these sort of new technology sorters come and go, there's always been drawbacks to using them, and most often they were issues with throughput or difficulty from an operator perspective, or they didn't have quite as many parameters that really stymied the ability of these instruments to really kind of influence the field at all. But this data, I think, indicates that normal types of sorting experiments can be done here, and you can get yields that are the same or better. We have a new high throughput cartridge that runs twice as fast as our first cartridge. So I think we're absolutely comparable, if not better, from a speed and yield perspective. Purity is absolutely achievable up to 99+%. You set up your experiment the right way. And the ability to sit and let it go without having an operator sit there is really transformative. And I hope that this is enough to at least put that idea out there. We have a lot of research that's been done, that's being done on this. I'll go into a little bit more on really the breadth of this technology to sort of validate that, okay, yeah, it's contained, yeah, it's not generating aerosols, but is it really as good as working with a traditional sorter? Can I really implement this into my workflow? And so what I'll show you here is information from an antigen-specific T cell sort that's been done as part of a therapy. And this cell sorting, I think the material starts at like 0.1% of the targets. Yeah, I'm realizing I didn't put that on this slide. But antigen-specific T cell sorts are always with very, very low starting targets. And they are able to pull up to 94% purity in a single sort. So these are, this is a workflow that's being implemented in a clinical trial currently. So I'll touch on a couple other things, but I want to come back to that point too. Lots of different cells have been sorted on this device. Obviously, when you come out with a new machine, you just have to get it out there and start trying a bunch of different things. But whenever this device has found its place into a core, that's a great way to get introduced to lots of different cell types. And what you'll notice is that a number of these examples have a variety of different sizes too. So iPS cells are usually quite large. T cells are, everything can sort a T cell, but bacteria will sort well, parasites, really, if you come up with it, we'll give it a shot. I think there's more and more evidence to show that this can be more of a Swiss Army knife than I think we expected. A lot of this talk has been geared around sterility and integrity of your sample and keeping that contained into a device. And I think operator safety is a huge part of that, but I think what's really validating to our ability to do that is the implementation of this device in the clinic or translational work. And in this context, we're integrating into a system where samples usually pulled from a patient, some sort of either viral transduction or enrichment of some kind, some sort of culturing occurs where we're either giving extra power to the immune system or culturing up the immune system so that it's more present in the tumor environment, and then going back into patients. And so to be able to get into the regulatory discussion of, is this product clean enough, sterile enough to go back into a patient, there's an extensive amount of qualification that has to go into that. And I'm excited to say that the Tyto has already been used in this environment. It's part of phase 1 and phase 2 trials actively right now. Over 200 patient samples have been sorted, over 50 of which have already been provided and infused into patients. And this is expecting to expand into even more trials this year and ongoing into the future. So but I think I felt it was necessary to include this into this presentation because I think nothing can speak more tightly to how enclosed, how clean this sample is and how defended it is from other cell types getting in there, from other contaminants getting in there, and therefore things getting out, than being in a clinical setting. And so I'm really excited about seeing where the cell therapy world takes this device. And I think it's just a really great example of just how enclosed this system is and how safe we really think it is for an operator. Yeah, I think I hope that this set of data encourages you to look at this device and think about it, how it might change your perception of doing cell sorting with material that are deemed hazardous. Again, I think this is a way to take this type of work into a place where you may not have the expertise right now. And we've already placed this new placements for people that are angling into the COVID research landscape, people that may not have access to an infectious disease core somewhere where they actually have the ability or the expertise to run in this environment. And I look forward to engaging with any questions you might have in response to this. I think something that I've learned about working in this job is I know a lot about our system. I know a lot about what we've done. I do my best to get informed about what questions people are asking, but until I interact with people, I don't know exactly how to support every single type of research. I think these types of engagements are really exciting to learn about what it is you're thinking about. And our team is dynamic enough that we're actively responding to questions. It's something that I think at Miltenyi, we have the luxury of being a private company, so we can kind of put our research into wherever Stefan Miltenyi sees fit. at the behest of our marketing team and probably our sales team too. But it's a company that's based really on innovation, and his kind of life's mission is to make cancer history. And so this context of COVID provided a really nice platform to demonstrate how quickly we were able to respond to questions in the field and address these issues of, oh my God, we'd love to work with COVID patient material, but we don't have the faculty to do that right now. So with that, I will wrap up. I've already kind of gone over the conclusions, but I'd love to take questions from you. But I have my email address right here. I'll put this disclaimer up. Yeah, I guess I haven't read through every detail there, but please reach out to me. My email's right here. My phone number is here. I'd love to answer any questions you might have on this, or see what I can pull from our company to help you. We have GMP materials, we have application data, we have an application development team if you're working on new materials. And also, reminder, please reach out to me if you want an invite to the webinar on the University of Chicago B-cell sorting for COVID samples, because I think that's going to be an awesome presentation as well. So with that, I'll sign off. Thanks for joining me for the not quite an hour, but maybe that'll give you a little extra time to grab coffee before the next thing. Yeah, thank you for joining me, and I look forward to hearing your questions, and I will hopefully talk to all of you soon. Thanks.